最初は生命科学分野で受賞されました米国ロックフェラー大学教授のデビッド・アリス博士に Beyond the Double Helix, Varying the Terrain of Epigenetic Landscape in Development Disease というテーマでご講演をいただきます。
So as you've heard, my background is really related to genetics, related to the genome. And what I'd like you to notice with these four pictures is that no matter which one is your favorite biology, whether it's fruit fly, eyes of different color, different mice, different sizes of mice, different colors of mice. Some of you know the female calico cat, very spotted with different coat color. And in this case, two identical twins who are not identical. One of these two sisters is, as far as we know, normal. The other sister is autistic and no names. And so it's interesting, I think, to consider that in all four of these examples, their genetics are identical, but not their outward phenotypes, not their expression, eye color, size, coat color, patches of fur, or in this case, a very complex psychiatric disorder. Their genetics are identical. So what's going on there? Of course, how exciting was it for the entire scientific community, a remarkably long time ago now, but it seems like yesterday, that the genome, the sequencing of the human genome was solved. Every letter of the DNA, every base pair of sequence was identified and reported in these two prestigious magazines in 2001. And how exciting was this for the worldwide scientific community? Well, of course it was exciting because the Human Genome Project taught us that genes, or we believe the logic, was genes determine disease, genes determine aging, and this is a big deal for me, the older I get from what you just saw, and genetic analyses would give us a window into diagnosis and possible therapeutic treatments. So all of these things motivated the Human Genome Project. And here's an example, a recent example. I apologize if you can't read every word, but that's not what I want you to pay attention to. For a fairly small amount of money in the United States, you can actually go to this company and learn about yourself through your DNA. And what might you be interested in? You might wonder what carriers of various traits you have. And let's look at sickle cell anemia, a red blood cell disorder that's frankly more inherited in the Afro-American population, but it's genetic. What about responses to drug? The hour is getting late. It seems like caffeine metabolism might be something we might think about. Biological traits. I think you don't have to be my kind of scientist to know that our genes determine eye color. And then, of course, maybe most importantly, disease risk. Whether you're female and perhaps worried about breast cancer inheritance within your family. For the men in the audience, prostate cancer perhaps is a worry. And of course, for me personally, longevity. I'd like to live a long time, and I wonder, I should perhaps go to this company and get the answers. And the company will tell you, the more you know about your DNA, your Watson-Crick DNA, the more you know about yourself. And that's pretty exciting. But there were some puzzles that were left unanswered by the Human Genome Project. First of all, why so few genes? only 20,000 or so genes make up the human genome. And that seems odd, especially when we think of ourselves relative to worms, flies, and fish, because they all have about the same number of genes, roughly 20,000. So how can that be? Certainly, we're superior to those organisms. And why are some of us so different from one another? Even the example of the monozygotic twins, the identical twins. Of course, how intriguing is it that we might be who we are based on past experiences in our environment, and that can't be explained necessarily by changes in the DNA. And if we're not selfish and worrying about ourselves, well, what about our children? Or in my last slide, my grandchildren. And then, you know, are there human diseases, are there human pathologies that cannot be explained at all by DNA sequence? The genes have been studied and no changes in the DNA have been found. So what's wrong? Where's the pathology coming from? So I'd like to call your attention to the real title of my seminar, Epigenetics. What is epigenetics, and why is it so exciting now to many in the scientific community? Well, it might help you to think about these simple analogies. If genes are words, epigenes may be how these words are read. Do you read a word, even in Japanese, do you read it softly? Do you read it very forcefully? That might be an epigene example, but the word doesn't change. It's just how you stress it. It's how you say it. Genes might be equivalent to a computer hardware where that's fixed, but you can alter what you do with that computer by the software programs that load into the computer. And you might think of the software for a computer as an epigenome or epigenetic analogy. And now, I think no matter what type of biologist you are, for some of you in the audience, you probably will find one puzzle piece here that will intrigue you. In my background as a scientist, I started out as a developmental biologist. That was fascinating to me. And it's interesting that I now have crossed through this epigenetic, which I'll explain molecularly in a minute, to even now a human disease puzzle piece. But you can see there's many other very interesting aspects that have been potentially linked to epigenetics. So, as I said, these are the two sort of Dave Allis background pieces where I wouldn't have necessarily thought epigenetics was going to be where my scientific journey took me. So historically, and I think history is important in science, epigenetics stands from or comes from the Greek prefix epi, stands for over, above, in addition to genetics, something on top of the genetics. And the person who's given largely in the community credit for in introducing the community to epigenetics is Conrad Waddington in 1942. Even I wasn't born yet. And this is one of his famous drawings where he was trying to depict an epigenetic landscape where this would represent clearly a ball, and this was going to be a fixed genetic unit, but whether the ball rolled down the contour map and landed here, or took a different path in development and landed here, was what he felt might influence genotypes as compared to where the ball finished, which were phenotypes. And in biology, we mean outward appearances. So think back about my first slide. Fly eyes, different color mice, calico cat, two different identical twins. And this was Waddington's concept. So if the DNA didn't change rolling down the hill, What did change? And now in much more modern terms, the Waddington epigenetic landscape is thought of this way. When sperm meets egg in development, one genome is created that ultimately will give rise to an embryo, multiple cell types, and as these cell types mature, different phenotypes, different outward appearances, different cellular identities are created. You might have red blood cells, you might have muscle cells, nerve cells, skin cells, and so from one genotype, how can you get so many different cellular types? And so clearly the genotype that we have only one of that's fixed has to be plastic. It has to be moldable. But some of you know that these cell types will divide once they're created. Not nerve cells, but everything else here will divide. So once the cells know who they are, how do they remember who they are? So there's something about this that's stable. The cells have been set up to give one particular cell type, and they know what they're to do for the rest of their development. But I don't think I need to introduce you to one of your very famous countrymen, and John Gurdon, who taught us there is a way to go backwards. You could take these stable phenotypes, and with the right set of conditions, you could reverse or reprogram or turn adult cells backwards into a more immature, pluripotent, Yamanaka factors. And I take my hats off to these 2012 Nobel laureates. And as a footnote, I want to tell you that both of them will be the 2014 honorary doctorate they will be receiving the 2014 honorary doctorate degrees from my home university, Rockefeller University in Manhattan, New York, this coming June. So we're very excited to have John Gurdon and Shinya Yamanaka receiving our honorary doctorate degrees this coming June. That's sort of a surprise. But maybe some of you know that. And unfortunately, if you try to reprogram cells and force them backwards, all is not always well. And the cells can become confused, and these cells can sort of lose enough of their identity that they take on a cancerous phenotype, which obviously is not a situation. This is what we want to avoid. And that, I think, is now one of the most important sort of topics. How do you distinguish going backwards with, say, for example, Yamanaka factors and induced pluripotency, IPS biology, and avoid a cancerous phenotype? So where my laboratory comes in is here. The human genome is packaged in a highly constrained, highly ordered mechanism, which many of you know and you heard in the introduction, requires a protein coat, a protein package. And those proteins that package our genome into these units, some people consider this a bead on a string necklace, like a pearl necklace that a woman might buy. They package the genome into higher and higher and higher structures that ultimately will fit into the nuclear space and even ultimately give rise to chromosomes that can separate during cell division. And some of these regions of chromatin are closed. They're shut off. They're very inactive. For example, the ends of the chromosome and the middle where the mitotic spindle attaches to pull the chromosomes, the sister chromosomes apart. These are sort of extra condensed chromatin areas known as heterochromatin. And in contrast, there's regions shown here in sort of green color that are much more open, much more permissive to gene expression. They're considered active regions of chromatin or euchromatin, truly active areas. And importantly, my lab set out many years ago to try to understand how heterochromatin or silent chromatin was different from active chromatin. Remember, the genome doesn't change. This is the same DNA. You're packaging it differently. And that seemed to us to be a formidable problem, given that the unit of chromatin is shown here, resolved in atomic structure by Tim Richmond and Carolyn Luger now some time ago. Shortly after the year that some of our work, which was mentioned, was published. So here you can see these beautifully colored histone proteins in the middle that are coming together to package a unit like a spool upon which the thread is wrapped. Where the thread is wrapped is the DNA on the outside, the Watson and Crick double helix. And these histones package a unit of chromatin, but the question is, how are they different between active and inactive? And here's where we took advantage of this tetrahymena model in my younger days. I'd like to show some of you that some very talented people have come into my lab and even given me amazing gifts. This is a cross stitch that was given to me by one of my former PhD students, that she actually sewed this gift, as you can see, to look like the crystal structure of the now famous Richmond and Luger nucleosome. But I think this is a beautiful gift that I'll have framed in my office, done by Lindsay Baker, who's now no longer in my lab. So all I really want you to really know about active on the bottom, open, extended chromatin versus condensed silent chromatin is that you can add different chemical marks to the histone proteins, and that's what my lab, I think, is best known for. And many other scientists worldwide are studying the differences between these two kinds of chromatin. So in green, I'm trying to depict a chemical modification that will tell the chromatin to open. And almost like a switch, almost like a light switch, there are different chemical modifications, shown here in red, that will condense the chromatin. And the question for us became, how do those chemical groups get added? Who adds them? And might that make any difference, really? And for us, that was kind of a gamble. And that took us to the discovery in 1996. I think this may be part of why I'm standing here before you tonight, to the enzymes that we've likened to those that add the chemical groups, so we'll call them writers. They add the group, or in contrast, enzymes that can take the group off, and we'll call those erasers. And if the particular chemical group was something that we call an acetyl group, this is really what I think my lab is probably best known for. Those enzymes that put on acetyl groups or take off acetyl groups. But in contrast, you can put on that red stop sign and have different writers and different erasers that remove these kind of silencing chemical groups. And this became for us a very enjoyable collaboration with groups in Europe, notably Thomas Genuine's group, who was at the IMP at the time. Notice that now we have 4 years difference between the discovery of these enzymes in 1996 and the discovery of these enzymes in 2000. And so this is probably apologetically too much detail for some of you, but let me take you through it for where the clinical connections come from. You might try to activate chromatin, so let me take you through it. You might use what we call a writer, a histone acetyltransferase, to put on an acetyl group. Now we know very well in the field that there are proteins and protein complexes, here marked X, that actually read those acetyl groups. They come and land on the acetyl groups. And if you decided that that was activating, if you needed to then silence that gene, you would turn to one of these histone deacetylases, an HDAC in short, to silence that gene. So that's the activation side of the coin. You add acetyl marks, you read acetyl marks, and when it's time to shut the gene down, you remove the acetyl marks. Now let me ask you to kindly flip this over to a silencing paradigm, a gene silencing paradigm. You can't add the methyl mark, the stop sign, the red mark, until you remove the acetyl mark. So here again, you see the importance of an HDAC. It takes off the acetyl group. That makes this particular amino acid in the histone ready to accept a methyl group. That's a histone methyltransferase. And now we know full well in the field that there are proteins that dock on the methyl. They won't dock on the acetyl. And when they do, the chromatin silences. And therefore, you have a silencing event. Now, importantly, in either of these two examples, the DNA has not changed. The DNA is identical. So what if in human disease, there has been mistakes made in silencing or mistakes made in activating genes? You might then think, I haven't changed the DNA, so let me now try to develop chemical inhibitors to inactivate these enzymes. And this is exactly what we and others did, and it became most fruitful to attack the HDACs first. And as an example, this is now a human patient that was diagnosed at Memorial Sloan Kettering, right across the street from where I am. This patient was in a bad situation with a metastatic cancer of the larynx that had now spread to this patient's lungs. You can see big, solid tumor masses in this individual, 2 different scans of the chest cavity. And after receiving treatment with these HDAC inhibitors, this is the same individual later, after 8 weeks of treatment. And in fact, these chemicals, these drugs, now became the first United States FDA-approved drugs for the treatment of various human cancers that ultimately became marketed and sold by Merck in 2006. But you can see, what looks to be a bad situation here by a much improved situation here. And I'm pleased to report to you that this patient is alive and well. And that, to me, is a very fulfilling part of the contributions that we and others have made. This is more graphic, and I apologize, but it's a good visual because it's an individual also with a skin cancer before treatment and after treatment. Again, also at Memorial Sloan Kettering. Very easy to grasp a large improvement in that individual following treatment. I'm very pleased to say that all of this success led myself and others to write a textbook in 2004. Of course, I have to admit, we wrote that textbook in English, but one of your scientific greats was kind enough to translate this book into Japanese, and I just now want to show you one figure from that book, which is a very heartwarming figure because for me it was such a stunning example of using these different chemical marks to do different important things. So this takes me to the laboratory of Shiv Grewal, and at the time, 2 of his young, talented Japanese postdocs who participated in the academic session we had the other day. And in this case, Grewal and colleagues literally asked the question, in one particular yeast genome, where are the silent genes? And the silent genes on this chromosome were all in this location. So using some tools from my lab, they first asked this question: where are the methyl marks? Where are those stop signs? Where are the silent marks? And I won't tell you the method, but I hope you can see in the actual experiment there was very strong signal here using some antibodies that really said these methyl marks are located here. Here's a graphic representation, but importantly, those methyl marks were not here and were not here. And when we switched, or I gave them tools and they looked at one of the other types of methyl marks that's activating, that's the on type, and did the experiment, I hope you can see down here that their actual data was very strong here, very strong signal here. I hope you'd agree with me, it's strong here, and it was very depleted, very reduced here. It's as though you indexed this genome from left to right with different active marks or inactive marks. And if this is a yeast, you can imagine what the human genome looks like when you index it with different chemical modifications. And that, I think, is really a picture worth 1,000 words in terms of using these chemical marks to index the human genome. This is way too much data, but in summary, whatever the chemical mark is, whatever it is, the enzyme systems that add them, subtract them, or the proteins that read them have now been identified by the scientific community at large, and I'm pleased to tell you that all of these different details are different active drug targets in the pharmaceutical industry. And I'll only say a few words quickly about one other situation, one other methyl mark here in sort of some of my closing scientific remarks. So I want you to remember something about a methyl mark at a particular amino acid. We call it lysine. It's abbreviated in one letter code with a K. And this is a very well-studied repressive chemical mark. So remember, or try to remember with me, lysine 27 methylation, and the enzyme that adds that chemical mark as a writer. And this takes me to another nice Japanese picture. These are two former members of my laboratory, two very talented MD, PhD scientists. They're actually a husband and wife. But it was Aaron Goldberg shown here, and what I want you to know is I didn't know they were coming to Japan, but they came to Kyoto on almost like a honeymoon and took this picture. And what I want to have you admire, besides this handsome couple, is their T-shirts, which are our new laboratory T-shirts that say, "Blame it on my epigenetics." So I think that's a very, I hope you agree. Doesn't seem like you agree. I hope you agree. This is a very cool T-shirt. But Aaron and Grace were visiting Japan, and Aaron's research project in my laboratory is what I want to turn to next. And that is the following. I described the histone proteins to you, but I never told you they can change. Not the chemical marks or the chemical tags, but the histone proteins themselves can be slightly different. And Aaron thought that would be a very interesting PhD project, and he went after a particular histone variant that happened to be in the family of histone 3. Too much detail, but I just want you to remember that this histone can go into the bulk chromatin, into active regions of chromatin, or even into silent regions of chromatin. And you might say that's impossible. How can the same histone go into multiple positions in our genome, in our DNA? And the answer was it uses different biochemical systems that Aaron and we and others studied. But never would I have thought there was a human disease connected to the third pathway. So when you take this particular histone variant and you put it into silent chromatin, I introduced that to you early in my talk, you have an important process that this paper, not from my laboratory, published in 2011 from primarily a cancer biologist at Johns Hopkins University, that's Baltimore, Maryland, and they reported high frequency of mutations in these two proteins. Now I'm going to go back a slide, I apologize. That's this protein, DAX, and its partner in life. And they found that these were high frequency mutations in a form of pancreatic cancer known as pancreatic neuroendocrine cancer. And I just will say, sadly, this is the cancer that Steve Jobs passed away from in 2011. So we were very fortunate in my laboratory to connect with these researchers, and now a very active project in my laboratory is to try to attack this cancer, knowing that a special histone is deposited by special machinery that seems to be mutated, and that's real genetics, in this disease. But I should be closing my remarks soon, or Toshi's going to give me a sign. And I want to tell you now about something I would have never imagined possible in my scientific career. I've been studying histones for a long time, but never would I have thought in my wildest dreams that they would be mutated in a human disease. And this is now probably one of my most active laboratory's current projects. That very lysine that I pointed out, I apologize if this is too much detail, in a form of pediatric brain cancer. Not any pediatric brain cancer, the most devastating pediatric brain cancer. This is a brain cancer that affects the brain stem. It's known for the experts in the audience as DIPG. It's diffuse, it's intrinsic, and it affects the brain stem known as the pons. And DIPG tumors, once the diagnosis has been made, the sad reality is these children are done for. Whether they're going to pass away in 1 month, 2 months, a few years, but right now it's essentially 100% death sentence in these children. And what was found by these groups, not mine, is that they have mutated the very amino acid that normally attaches these chemical groups with remarkable frequency, in some cases 80%, to another amino acid, methionine. And here's an example of human autopsy pediatric tumors stained with an antibody from our lab. These were children that passed away where they don't have this mutation, but they had other mutations that caused their death. So I hope you can see from even the back row, there's a lot of staining. A lot of these nuclei are stained darkly. But 100% of the children that pass away, that succumb to the mutations where they have been sequenced to be K27 to this amino acid methionine, no staining whatsoever. No staining. For those of you that are critical scientists, you'll say, oh, I see a little staining here. This is a non-tumor invasive vascular tissue. This makes a nice positive control. But I hope you would agree, very strong staining here, no staining here. That seems impossible, And last year in my laboratory, Peter Lewis identified the simple finding, but this is a breakthrough for this field, that this simple mutation, seemingly simple mutation in the histone protein is completely inactivating the writer. That's the only reason I took you through this detail earlier. It poisons this methyltransferase so remarkably that you see no more of the end product signal whatsoever. So what's our goal now in the laboratory? Now our current focus in the laboratory is to learn how to detoxify this mutation. And we're making some progress on that, and I think that's going to become one of the most exciting sort of future avenues of research for my laboratory and others who study this devastating childhood cancer. And I really hope I can come back and tell you about progress in this area, because right now there is nothing good to report for these children. So to conclude, almost, this has sort of been the timeline of my career. It sort of tells you when different, in time, when different key enzymes were identified by us and by others, even leading us to the current sort of example of mutations in the histone proteins. And at the end of the day, we feel that what that does is throw a balance out of whack. Normally, cells want to grow properly. They want to take on their properties and differentiate. But when some of these epigenetic enzymes become dysfunctional, dysregulated, it seems as though this balance tips towards one of very aberrant cell growth, de-differentiation, and cancer ensues, tragically. This is a boring list, but now it's just not cancer alone. When physicians have now administered various other agents to treat various other disorders, I think you can read this list as well as I can. In English, various diseases that a lot of us can relate to: inflammation, perhaps depression, hypertension, pain. In all of these cases, it seems as though it's the epigenetic layer that's become the problem. And now this is very exciting. I just want to leave you with the message: it's not cancer alone that epigenetics is being called into play. And this is not my graft. I would never do this. But Reuters published a simple citation curve on the word epigenetics, and I just sort of, I'll take you through some high points. The enzymes that my laboratory is being recognized for here today with this phenomenal JapanPrize were discovered in 1996, methyltransferases in 2000, demethylases in 2004. Some of the readers of these modifications were chemically drugged successfully in 2010. The mutated histones were discovered off the chart in 2012 and 13. I told you that's where we're focusing our attention now. So I don't know what the future holds, but I have a feeling it's going to keep going up. And these are now numbers of papers, numbers of proteins that fall into this category, clinical trials that are underway in the United States and worldwide. This isn't just biotech companies, this is big pharma. And it leads me to a quotation from Jim Watson. Jim Watson, you know, is the DNA molecular Nobel Prize winner with Crick, Francis Crick, for the discovery of the DNA structure, the double helix. And when he writes, you can inherit something beyond the DNA. This is my take-home. These are his words, not mine. He says that's where the real excitement is now. But certainly, it's in his mind, he's thinking about something that you can inherit. That should make you think about mom and dad. That should make you think about inheritance, your parents. But this was one of my opening slide examples of biology. These are all genetically identical mice. But guess what? what happened that was different in these mice is it's what the mother ate. The mother ate a different diet that was containing different methyl group donors. This is unbelievable. So these are the children that are genetically identical, but the mother ate a different diet. Not our work, but that's remarkable. Some of you might say, oh, but this is mice. I certainly don't care about mice, but I'll sort of conclude my talk by saying that this article made the cover of a United States popular magazine, Time magazine, and it really was paying credit to this physician from Sweden who very meticulously tracked, and I should say this is his son, this is his grandson, and they lived in the most northern portion of Sweden, and as a pediatrician, he tracked the health of individuals under his care when this part of Sweden was either very, very rich in agriculture or periods of time when it was really very poor in terms of agriculture due to harsh winters and so on. And what he found was a remarkable correlation in these individuals' weight, lifespan, disease susceptibility, and even mental health. And he's given credit now for something about diet, something about feast or famine in this very northern portion of Sweden that cannot be explained by DNA changes, must be explained by something else. So I have 3 slides to leave you with. They're the same. They're the same, so don't panic. I'm really almost done. This is what we all grew up with. I think it's a beautiful slide. It says it's DNA. DNA is the key to our genetic inheritance. And we can turn genes on or off by various factors that bind the DNA. Notice, this even looks like it's naked DNA, just Watson and Crick DNA double helix. This is true genetics. This can really not be changed much. It's fixed. But I hope tonight, in my lecture, I've convinced you that we also have to think about another layer. And that's these chemical modifications that can either be added to the DNA itself, some of you know this is methylated DNA, or to the histone proteins that could be methyl groups, could be acetyl groups, but the important part is that this is something in addition to the DNA. And it's been shown by the community at large that this chemical states of chromatin can be inherited. It's epigenetic. It's much more responsive to changes in the environment. It's very dynamic. And this is now what I think people are excited about. This may be what Watson was referring to when he said in his quote, you can inherit something beyond the DNA. And that's one thing that I think we all are going to worry about, because whether it's something about cigarette smoking, something about too much sunlight, perhaps our diet, what we eat, yes, you can change the DNA by mutation, but it looks like much more is happening here that I hope I've intrigued you with tonight in my lecture, the epigenetic side of the coin that's very responsive to these kinds of influences. And should we be able to pass these on to our children and our grandchildren, this is very exciting, a new area for the community at large. So what I want to sort of conclude with is I think one of the most special things about science is the people part. Maybe I'm just getting old, but this was a wonderful former postdoc in my laboratory from Japan, Reiko Oba, and when she left the lab, never, ever would my wife or I have expected she came to us with this gift. And the gift was this painting. I apologize, it's not a good quality. I took it right off of our bedroom window, our wall. But her mother, whom I've never met, painted this gorgeous painting for us as a small gift to us for the time that Reiko had been in my laboratory. this was one of the first amazing tastes I had of your culture. I had never received something so beautiful. My wife and I are truly honored to have this over our bedroom, master bedroom wall. And I think for me it sums up what I think is so special about your culture. I'm not sure anybody in the United States could make something so beautiful. And it just represents, I think, your generosity and kindness, which we've certainly seen today. These are current members of my laboratory and a few past members of my laboratory. Much of what I've talked to you about is done collaboratively, and I just want to point out that many of these persons in this block of my contributor list are physicians who are in our neighborhood in New York that are kindly getting us, various human disease tissues to work from. And these are some of our funding sources. And perhaps most importantly, without all the clutter, although this is the contributions, I'd like to absolutely say to you, to everyone related to the JapanPrize Foundation, to everyone this week that's treated myself and my family so warmly, thank you so much. I am greatly honored to be here and to receive this prize. Thank you so much.